I'm Jane Morris, and in this presentation I'm going to talk about genetically modified nutritionally enhanced crops, otherwise known as biofortified crops, with particular reference to their potential application in southern Africa. I'll first address the question of why we need to use genetic modification to biofortify crops, rather than relying on other techniques to address nutritional deficiencies. Then I'll talk a bit about the crops and traits that are in the pipeline, and finally discuss the risk assessment of these crops and also what are the hurdles to introducing them into the market. Let's look first at the problem that we're trying to address. Well, around the world we know that 2 billion people have inadequate intake of essential micronutrients. The problem is particularly serious in southern Africa, where 40% of children under 5, that's 18 million children, suffer from malnutrition and stunting. This has long-term effects because it impacts on their health, learning ability and economic potential for the rest of their life. Programs to provide vitamin and mineral food supplements have limited reach and aren't fully overcoming the problem. While there are efforts to fortify staple foods such as maize meal, this requires coordination and a distribution infrastructure, and it's not going to be applicable for many small-scale farmers. Just to emphasize the scale of the problem, this map shows the percentage of the children under 5 in each country that suffer from stunting. Even in South Africa, the rate is 27%, and ranges up to 47% in Malawi and 50% in Madagascar. This is not just a tragedy on an individual level, but will affect the whole country's ability to develop economically. It puts considerable pressure on scientists to develop new approaches to biofortification for a range of essential nutrients. So, what are the possible ways of addressing the problem? Biofortification can sometimes be achieved by adding nutrients to fertilizers, an approach that has been very successful to increase the availability of zinc and selenium. However, this is not suitable for iron because it's immobilized in the soil, or for essential organic nutrients, that is vitamins, essential amino acids and fatty acids, which are the products of plant metabolism. In any case, in developing countries, farmers do not always have ready access to fertilizers. It may be possible to increase the content of organic nutrients by conventional breeding if there is sufficient genetic diversity in existing germplasm, although it would take many generations to introgress these traits into local elite breeding varieties. On the other hand, the GM approach offers not only an unrestricted choice of genes for nutritional improvement, but also has the potential for nutritional traits to be targeted to specific organs, such as cereal seeds. And multiple traits can be combined in the same plants without complex breeding programs. So, what nutrients are the main focus of GM biofortification efforts? Starting with the vitamins, the main focus areas are vitamins A, C, B, that is folate, and vitamin E. Vitamin A is essential for eyesight and helps maintain healthy epithelial and immune cells. Vitamin A deficiency is the most prevalent form of deficiency in developing countries, and the prevalence in sub-Saharan Africa as a whole is estimated at 48%. Vitamin C deficiency is becoming increasingly common. It's an antioxidant and also cofactor of several important enzymes, and plays an important role in early brain development and chronic disease prevention. Folate is essential for DNA synthesis and many other core metabolic reactions. Its deficiency causes anemia, but is particularly important for pregnant women, and a shortage can lead to spina bifida in the fetus. Unfortunately, there's not a lot of data on the prevalence of folate deficiency in Africa. Vitamin E comprises 8 related molecules. They are powerful antioxidants that protect fatty acids, low-density lipoproteins, and other components of cell membranes from oxidative stress. Vitamin E deficiency is common in developing countries, and it's exacerbated by infection with malaria or HIV. Moving on to the essential amino acids. 9 of the 20 amino acids are essential nutrients because they can't be synthesized by humans. The most relevant examples are lysine, threonine, tryptophan, methionine, and cysteine, because staple cereals are poor sources of lysine and threonine, and staple legumes are poor sources of tryptophan, methionine, and cysteine. Lysine is the first limiting amino acid in a maize-based diet, so it's been a particular focus. As an alternative approach to increasing the levels of individual amino acids, scientists have also tackled the problem by introducing elevated levels of storage proteins rich in a range of essential amino acids. Humans can synthesize most fatty acids de novo, but the health-promoting omega-3 and omega-6 polyunsaturated fatty acids are exceptions that need to be sourced from the diet. They're critical to brain development in children. They're abundant in fish, shellfish, nuts, and leafy vegetables, but they're not present in cereals, so this can be a problem in southern Africa where many people don't have access to a varied diet. Unlike organic nutrients, which are the product of plant metabolism, mineral nutrients must be taken up by plants from the environment. The most significant minerals in terms of nutrient deficiency are iron, zinc, selenium, and calcium. Then, in addition to increasing the content of key nutrients, another focus is to reduce the content of anti-nutrients. Phytic acid is a key anti-nutrient because it's abundant in cereals, legumes, and oilseeds, where it binds all the principal mineral nutrients and sequesters them into stable complexes that can't be absorbed. Lectins are carbohydrate binding proteins that are found particularly in beans, but also in cereal grains, seeds, nuts, and potatoes. They're toxic if the food is consumed in uncooked or improperly cooked form. Adverse effects may include stomach upsets and allergic reactions. Cassava is an important staple crop in sub-Saharan Africa, but the high content of the cyanogenic glycosides, linamarin and lottostralin, means it must be processed before consumption. The levels of these toxicants are not always adequately reduced by processing, and there are a number of instances of acute and chronic symptoms of toxicity. It clinically manifests in central nervous system and cardiovascular disturbances that could result in coma and death. In addition, consumption of cassava and its products can result in konzo, an irreversible paralysis of the legs, and tropical ataxic neuropathy, a chronic condition noticed in elderly persons. Goiter and cretinism could also be aggravated by cyanide from cassava, especially in areas of endemic iodine deficiency. Scientists are working on quite a wide range of crops, as shown here. The most well-known is the Golden Rice Project that has been ongoing for a number of years. Of particular relevance to the southern African region are maize, cassava, and sorghum, although the African Biofortified Sorghum Project seems to have stalled, unfortunately. There are a number of ways in which scientists are working to use genetic modification techniques for biofortification. One way is to increase the flux through a biosynthetic pathway, for instance by increasing availability of precursors or removing feedback inhibition. A similar approach is to overexpress biosynthetic enzymes in a pathway. Then, if the pathway is missing altogether, there's the possibility to introduce a complete new biosynthetic pathway. For essential amino acids, one approach is to get the plant to produce proteins with better nutritional composition. GM approaches to enhance mineral nutrients are quite diverse. For iron, one strategy is to increase its solubility in the rhizosphere, for instance by overexpressing reductases to convert insoluble ferric iron to soluble ferrous iron in order to increase uptake from the soil. Other strategies include mobilizing minerals in the plants, transporting them to storage organs by increasing expression of transporter proteins, increasing the storage capacity of the plant, and maximizing bioavailability. expression of a thermostable recombinant fungal phytase increased iron bioavailability in wheat and maize. Then, for antinutrients, the goal is generally to reduce their production by silencing biosynthetic genes. As an example, the amount of phytic acid in seeds can be reduced by silencing genes involved in its biosynthesis, such as myoinositol phosphate synthase. For cassava, 2 genes encoding cytochrome P450s catalyze the first step in linamarin and lottostrolin synthesis, and suppression of these genes has reduced linamarin by 94%. RNA interference targeting the same genes reduced the cyanogenic glucoside content by 99%. I already mentioned the Golden Rice Project, which is a good example of a project to tackle vitamin A deficiency. Around 250 million preschool children are estimated to be vitamin A deficient worldwide, including many in Africa. Rice plants have the full machinery to synthesize beta-carotene, precursor of vitamin A, but while the pathway is active in leaves, parts are turned off in grain. Expression of 2 genes, a maize phytoene synthase and bacterial carotene desaturase, turns the pathway back on in the rice endosperm. Golden rice has not yet been released commercially, in part because of pressure from anti-GM activists, but also because yields have not been consistent, emphasizing that agronomic performance is as important as nutritional content. One example of a biofortified crop that has been approved for commercial release in the US and Canada is Monsanto's MON 87705 soybean. It's also been approved for food and feed use in a range of countries, including in the European Union. This soybean has lowered levels of the saturated fatty acids palmitic and stearic acid, and also the polyunsaturated fatty acid linoleic acid. At the same time, it has higher levels of oleic acid. These changes were achieved by downregulating 2 genes involved in fatty acid biosynthesis using RNA interference. Chemical hydrogenation was adopted to reduce the concentrations of polyunsaturated fatty acids and prolong shelf life of soybean oil. However, nutrition research showed that the trans fatty acids created by the hydrogenation process had negative health consequences. Many consumers are attempting to avoid trans fats, and as a result, there has been a decline in soybean oil consumption. By reducing the polyunsaturated fatty acid concentration, chemical hydrogenation can be avoided. Low saturated fats and high oleic acid levels are also key attributes for vegetable oils targeted for biodiesel and in industrial uses. These characteristics are important to improve cold weather performance, improve stability, and reducing nitrous oxide emissions. So, MON 8705 can serve a dual purpose for both food and feed use, as well as for industrial purposes. Another example of a biofortified crop that has been approved for general release is LY038 maize, also produced by Monsanto. Maize is particularly deficient in the amino acid lysine, and lysine is often used as a supplement, particularly in poultry feeds. LY038 maize contains a bacterial gene from Corynebacterium, encoding for dihydropicolinate synthase, which is the rate-limiting enzyme in plant lysine biosynthesis. The bacterial gene is more than 50 times less sensitive to feedback inhibition than the plant gene. It was also later shown that the inclusion of an RNAi sequence could increase lysine production in the endosperm by reducing lysine catabolism. The level of free lysine is increased from less than 100 parts per million to more than 4,000 parts per million, but at the same time there's no change in the protein-incorporated lysine, which is the major contributor to the lysine content of conventional maize. However, the increase in lysine content is sufficient to avoid the need for additional lysine supplementation in animal feeds, which is the main target for use of this particular maize variety. In general, the approach to risk assessment of biofortified crops is similar to that of first-generation GM crops where input traits have been modified. However, there may be some need for adaptation of the approach to risk assessment, considering that there is now an intention to introduce major changes in composition. Where the compositional alterations don't fit into the range of natural variation of the crop, the impact of the changes needs to be well understood. Particularly if the metabolic pathway or pathways involved are not fully understood, there may be a need for more scrutiny than usual to determine the effect of the introduced changes. Then, where there are major changes, it may be inappropriate just to have as a comparator a near isoline of the GM variety, which is the normal recommended practice. It makes more sense to consider the full extent of natural variation of crop composition to contextualize the changes. The impact on nutritional status will need to be determined, taking into account the range of dietary intake levels. And if there are changes in processing requirements or nutritional characteristics following processing, these will also need to be taken into account. The bottom line note, though, is that it will be essential to assess the benefits as well as the risks to ensure that the nutritional benefits are taken into account in reaching a decision. So, the general approach to risk assessment of a biofortified crop would go along these lines. First, you would determine if the expressed proteins or altered metabolites have a history of safe use. If not, then it would be necessary to obtain information on nutritional and toxicological properties of the expressed trait. Agronomic, phenotypic, and compositional characteristics of the crop that may have changed, either intended or unintended, need to be determined and checked to see if these are within the range of natural variation. If they're not, then further testing may be necessary. Next, you would assess the level of toxicants known to be characteristic of the host and donor species and check on the possibility of transfer of food allergens from the donor. You will need to check on changes in concentration and bioavailability of important nutrients. The safety and nutritional value of newly introduced proteins, as well as the composition and nutritional value of any other new or modified nutrients, needs to be determined. It may also be that different growing conditions will affect the levels of nutrients, so this needs to be determined. And finally, the influence of processing and storage on the characteristics of any derived products needs to be determined. When it comes to estimating intake levels, it's important to estimate how much of the food is likely to be eaten and how often. Perhaps food preferences and food availability will change as a result of the introduction of the GM food, leading to changes in consumption levels of a range of other foods as well as of the GM food. So then, the likely impacts on dietary intake of all nutrients, not just the introduced nutrients, need to be estimated, as well as the likely impact on intake of anti-nutrients and toxicants. And this all needs to be estimated in the context of particular groups, especially those who are particularly vulnerable, such as children, the elderly, or those with particular health or dietary issues. Then we come to the issue of labelling. Labelling can be regarded as more important when there are modified output traits that affect the consumer than when only input traits have been modified. Producers want to label in order to capture the benefit for the consumer, and they can also help consumers make informed choices about consumption. Because of the need for specific labels about the nutritional characteristics, it's likely that identity preservation systems might need to be put in place to segregate the nutritionally enhanced crop from other crops. It's also likely that there will be a need for post-market monitoring of nutritionally enhanced crops. It will be necessary to determine whether the anticipated health benefits are actually being realized at a population level. Where a component may be close to maximum daily allowance levels, the actual levels of intake need to be determined. There may be changes in dietary preferences as a result of the introduction of the GM crop, so the impact of any changes in intake need to be considered as well. And then there may be changes in the food processing methods, and the effects of these may also need to be determined over the long term. One very important aspect is the communication of risks and benefits. Up to now, most GM crops with modified input traits have affected the farmer, but have not had a direct effect on the consumer, except in terms of personal preference. At this stage, there is limited information on whether consumers will be prepared to accept nutritionally enhanced crops. Studies have been done in a few countries, but they show considerable difference between countries, and there's not enough data from Africa. Certainly, consumers will need to be aware that the products are different from those already on the market, and they need to understand the implications in order to make informed choices about consumption. Because the introduction of nutritionally enhanced crops will need to involve all players in the food chain, the stakeholders all along the line, from the seed suppliers to the farmers, the food processors, and then the consumers, will need to be involved. And because there's not necessarily a direct benefit to the farmer to grow biofortified crops, it may be necessary to provide them with an economic incentive. This may require political involvement. So, the bottom line is that the introduction of biofortified crops is going to be quite a complex process, and many hurdles remain if they're going to be accepted not only by the consumer, but also by other role players in the food chain. For this reason, a proactive approach is needed to address both regulatory hurdles and public acceptance. And finally, as I said earlier, assessment and communication of the benefits, rather than a focus merely on risk assessment, is essential. From that perspective, you might wish to have a look at the video in this series on risk-benefit assessment. In this final slide, I give you a few suggestions for further reading. The first three are papers from the open literature, while the last one is a report that was commissioned by the European Food Safety Authority to guide them in knowing how to approach the risk assessment of GM foods with modified nutritional content. I hope you find these useful. Thank you.